Back. So now it's the second part about our lab 9. So in the first part, we have already go through the basic concepts about the hidden Markov model and also the key questions the hidden Markov model is going to answer. So now I'm going to lead you guys through some examples to get in touch about the actual application. Well, before we do so, just please install the required package of the hidden Markov model in our session, which is the HMM. Well, it's very useful. So briefly, I would try to throw you guys a black box first, which could do all the remember all our algorithms. So we have touched the two type of questions, and we have talked about four algorithms, including the forward procedure, backward procedure, forward-backward algorithm. Well, if you want to conduct the forward-backward algorithm, you have to get the result of the forward procedure and the backward procedure, and also the Vitary algorithm. So how could we do all of these four algorithms in R? Well, R has already created a package as the HMM, so it's a black box. And then I would take you guys to calculate them by hand. So maybe through this part, you could write the whole package by yourself. Then okay, so now let's get through the examples first. Well, the first example we have encountered them in the first part is about the weather, and based on the weather to predict the actual activities of some specific guy on some specific day. Well, the setting would be that we have chances that on Monday, Tuesday, and Wednesday, we got different chances of observing a rainy day and sunny day on that specific day. While on a rainy day, the guy might have higher chances of cleaning their house. Sorry, I was too crazy about the data. Higher chances about cleaning their house, and on rainy day they have lower probability of walking outside, which is 0.1. And on sunny day, the chances of walking is 0.6, the chances of shopping is 0.3, and the chances of cleaning house is 0.1. Well, and then from rainy day, there are some changes between the two states. So rainy day could change between rainy day. So given that the first day is rainy day, the chances that the second day is rainy day is 0.7. And given that the first day is rainy day, the chances that the second day is sunny day is 0.3. And given that the first day is sunny day, the second day is sunny day is 0.6. And given that the first day is sunny day, the second day is rainy day, the chances that is 0.4. So remember that whenever you encounter a hidden Markov model question, you have to count about four elements first. So can you think about the basic four elements of this specific question? Okay, so it's example 1, weather and activity. So now the first state is so what's the first element? It's the states, right? So we have the states as the, if we can define the states as the states, so the state might be state in what the chances are raining or sunny, right? Yeah, so this is the basic events of the states. And what's the second part? So the second element might be the observations. So we might define them as a big O, which is this is not a big O. This is a big O. So observation. So how many observations do we have? Walk or shop or doing clean. Okay. Then the third element might be the transition probability. So the transition probability is the probability between different states or between the same states. So we have 4 transition probabilities, right? So now the transition probability from raining, the second day is raining, given the first day is sunny, is what? So it's this one, 0.4. And we got the similar one as transition probability. Given that the first day is sunny and the second day is sunny is what? So it's 0.6. And also the transition probability that given the first day is raining and the second day is sunny is what? It's 0.3, right? And also the transition probability that given the first day is raining, what's the chances that the second day is still raining? So it's 0.7. All right, so this is the third element. So what's the fourth element? The fourth element is the emission probability, which is the probability that connects the specific states and some specific observation. So, for example, given that the first day is given that the day is raining, what's the chances of being walking? So the chance is 0.1. And also given that the first day is raining, what's the chances of being shopping? So the chances is 0.4. And similar things for that, given the chances of given the day as a raining day, what's the chances of being cleaning, right? So the chances is 0.5. And then you can do the same thing for the emission probability of given the states as a sunny day. Sunny day go shopping, sunny day go cleaning. Sorry for this confusing part. And this one should be what? So given that this day is sunny, the chances of being walking is 0.6. Given that the day is sunny, the chance of being shopping is 0.3. And given that the day is sunny, the chances of doing cleaning is 0.1. Okay, cool. So that's the emission probability. So now you have already get all these 4 elements. So how could we set up all these 4 elements into the R session? So we have to do some startup issues, and all you have to do is to calculate all things you have to calculate are summarized in this table. So I call it as the all-in-one table. So with the forward algorithm, you could get all these alpha values about these 3 days. So we just want the predictions about Monday, Tuesday, and Wednesday. And with the backward algorithm, you can get all these beta values. And with the forward and backward algorithms, you can calculate this gamma. And based on this gamma, you can make the prediction at each state whether what type of weather are there. So now the question is described as given that we have observed this guy as walking on Monday, cleaning on Tuesday, and shopping on Wednesday, what's the prediction you would get for the specific weather of both Monday and Tuesday as well as Wednesday? All right, and now if you can open our session. So now you already know all these basic elements. So now we could try to define all these elements in R. Oh, sorry. All right. Before we do so, install the required package and load these modules. Then we can define the initial probability. So recalling the question, to begin with, we have a 60% chances to find the first day as a rainy day, and 40% chances to find the first day as a sunny day. So this is the initial probability. And then we could define the transition probability, which is this part as the third element here. So it's a 2 by 2 table. And then we could define the emission probability, where it's a 2 by 3 table. So it's the first element listed here. You can match each number here from 0.1 to 0.1 here. So actually, you can compare them one by one. And then we can prepare the basic data structure for the hidden Markov model. So now we could show the basic structure of the hidden Markov model. So basically the states we have raining and sunning, which is states here, and the symbols, which are the observations. They are working, shopping, and cleaning. And also the start probability, which is the initial probability, as 60% for the first day as raining and 40% for the first day as sunny. So just so then and the transition probability, which is the third element here, and also the emission probability, which is the fourth element here. So exactly the same. And then now our problem is that given that the observations as working, you can change the, you can play around and change the observation result. So now my question is, given that I followed this guy through Monday to Wednesday, and I observed him as working on Monday, cleaning on Tuesday, and working on Wednesday. So how would you predict the weather of Monday, Wednesday, Tuesday, and Wednesday? So now we could do this by using the hidden Markov model. Since the model is calculating a log probability, you have to do the exponential transformation. So first we should do the forward algorithm and calculated this alpha values, and then do the backward algorithms to get the beta values, and calculated the gamma value with the command as the posterior followed by the hidden Markov model and also the observations. So then, based on the posteriors, it gives you the chances of being sunny on Monday, being sunny on Tuesday, and being sunny on Wednesday, and also chances of being raining on Monday, being raining on Tuesday, and being raining on Wednesday. So based on these results, for example, on Monday, the chances of being sunny is higher than the chance of being raining, so we would predict the first day as sunny day. And on Tuesday, which is the second day, the probability of being raining is higher than the probability of being sunny, so we might predict the second time as a raining day. And for the third day, the probability of being sunny is higher than the probability of being raining, so we might predict the third day as the sunny day. So our results from the forward and backward algorithm might be what? So it's sunny, raining, and sunny. All right, so let's see whether we could get different results from the Viterbi algorithm, where R could also do the Viterbi algorithms directly. Well, the only difference thing between the Viterbi algorithm and the forward and backward algorithm is that they try to maximize different things. The forward and backward algorithm tries to maximize all these probabilities on each step, but the Viterbi algorithm just remembers the paths between different states. So we could do the, so we can see that for this specific setting, both the Viterbi algorithm and the forward and backward algorithm have get the same answer. Well, there are circumstances where they would produce different answers, but the chance is very small. I would say that the longer the length of the hidden Markov chain, the higher the possibility that you might observe different answers from the Viterbi algorithm and the forward and backward algorithm. Well, what if we can change the observations as walking and walking, or working on these 3 days? So now we can do these things again, and do the backwards to calculate the beta again, and calculate the gamma values again. So now you can see that we might predict all these 3 days as sunny days. So how could we do, what's the result about the Viterbi? So cool, they also got the predictions for all these 3 days as sunny. Great, so that's the first question. And then The second question is quite similar to your homework. Well, actually, just pay attention to these numbers. So I selected different numbers for the lab material. Well, for homework, I think your number for the initial probability might be slightly different. So just do not use exactly the same code which I used for the lab material for your homework. You would make some mistakes. Pay attention to these numbers. Well, so for this homework, it's talking about the CPG island, and given that specific CPG island, the probability of observing specific amino acids might be slightly different. So based on this background, if we have a specific sequence of some amino acids, such as the AGGCGT here, we can use the hidden Markov model to help us make predictions about the CPG islands or non-CPG island at specific loci. For example, whether this amino acid A coming from a CPG island or non-CPG island, whether this amino acid G coming from a CPG island or non-CPG island, and so for the remaining amino acids. So remember that the first thing we have to do when we are dealing with a hidden Markov model. Now it's the fourth example. It's the second example, sorry. So it's CPG island and amino acid. Alright, so the first thing is still to find the basic 4 elements. So what's the state now? It might be whether this specific loci is in what? CPG island or non-CPG island. So I would encode them as C for CPG island or N for non-CPG island. And so what's the second element? So it's observations, right? So for this scenario, the observations might be the actual amino acids. Well, I think for currently we have get 8 amino acids, right? There are 4 new members just since the beginning of the March. But for this question, we still keep the classical 4. So sorry for the type of. So it's observation A, G, T, C. Alright, and then it's the probability, the transition probability. So the transition probability is the probability that connects the different states or the same states. So now, if we can adopt this one, so the transition probability is between the CPG island or the non-CPG island and the CPG island, and also the non-CPG island and the alternative one. Alright, so if we can go back to this question, the transition probability between CPG island and CPG island, which means that given that the first loci is within a CPG island, the probability that the second loci is also in a CPG island is 0.7, so which is the chance here. And based on this number, we could calculate the number that given that the previous loci at the CPG island, the chances that the second loci is within a non-CPG island is actually 1 minus 0.7, so it's 0.3. And then the second one number that given that the previous loci is non-CPG island, the current loci being a non-CPG island is 0.6, right? Yeah, so it's 0.6. And given this number, we could calculate that given the previous loci as the non-CPG island, the current loci as the CPG island, the chance would be 0.4. So that's the transition probability. And the fourth element would be the emission probability. So the emission probability might include for example, given that this loci is in CPG island, the chances of observing an amino acid A is what? Is 0.1, right? So it's here. Cool. And you can do the similar thing for all these for all possible amino acids. So it's A, C, G, and T. And the chances are 0.1, 0.4, 0.4, and 0.1. Cool. And you can do the second half of the emission probability. Given that this loci is within non-CPG island, and now the chances changed to 0.3, 0.2, 0.3, and 0.2. Alright, sweet. So these are all the basic elements about this specific hidden Markov model. And now you have already know how to use R's black box to conduct the forward algorithm, backward algorithm, the forward and backward algorithm, and also the Viterbi algorithm. But now I would lead you guys to calculate all these details by head. So how could we do so? If we'll have to calculate all these numbers in this table, and you also have to report them in your homework. So basically, I have already written some specific details in the R session. So to begin with, we got the initial probability as 0.4 and initial probability that the first loci as the non-CPG island as 0.6. So if you want to explain them more clearly, actually you can drop some pictures. So we got the initial probability that the first loci as a CPG island is 0.4. And we also got the chance that the first loci as a non-CPG island is what is 0.6. Okay, and we observe some specific sequence as what? As AGGCGT. So it's A. And also the transition probability from a CPG island to another CPG island is 0.7. So similar things might happen for the C, and it could also have the transition probability between these two are 0.1. So it's 0.6 between different non-CPG islands. There are also chances that it might change from a CPG island to a non-CPG island with the probability as 0.3 here. And also the chance of changing from a CPG island to a non-CPG island as 0.4, right? Cool. Okay, and then our observations are A, G, G, C, G, T. Great. And then given that specific loci is a CPG island, the chances that this loci is an amino acid would be 0.1, which is this one, right? And given that the specific loci is within non-CPG island, the chance that this loci has amino acid at A is 0.3. So these are the emission probability. Okay, and then with this specific graphic interpretation, you can do the same thing that Shirley did in the lab, Shirley did in the lecture. If you have problems, please come to my office hours and I can give you detailed explanations about the graphic interpretation. I think that would help a lot to help you understand what we are doing for calculating the details about all these numbers within this table. Okay, so now we can calculate all these numbers. So this is the alpha 1 at the first at the first time point, and also time point alpha 2 that inherited alpha 1 from the first time point, and also alpha 3 that inherited alpha 2 from the second time point, and did the same things for all 6 time points. And then we can print all these alpha values. So this is the first row in this table, and then calculate the alpha values for the non-CPG element, which is the second row within this table. Again, please remember that the number is different between the lab material and the homework. Then we can do the backward algorithm to calculate all these betas. So it's similar things, but just begin from the very end at the 6th time point, and inherited the chances that we calculated in the 6th time point to calculate the probability of the 5th time point, and so for the 4th, the 3rd, and 2nd, and the 1st, and then print all these betas. So this is the row for the third row. So this is the third row in this table, and then this is the fourth row in this table. You have to report all these values in your homework, and then we, based on this beta and alpha values, we could calculate the gamma values, which is listed here. So how could we check whether our forward and backward calculation results are similar? So we can do the termination step. Remember that the termination alpha and the termination beta should be the same, exactly the same to each other. Well, this point of 4 is actually the initial organization probability that the first state being as the CPG element. This is number here. And the 0.6 is the initial probability that the first state as the non-CPG island. And 0.1 is that, remember that at the first state, we have observed at A, so it's the chances that, given that the specific amino acid as A, the chances that it's coming from a CPG island would be 0.1. And the 0.3, it means that, given that the first amino acid is A, the chances that it's coming from a non-CPG island is 0.3, which is 0.3 here, and also 0.3 here. Great. You need to use that to calculate the termination beta and to check whether these two are equal to each other. Well, in your homework, you might observe that there are tiny difference between these two termination values, but you can, if that thing happens, you can just type the termination alpha and the termination beta to see whether the difference is very small. And if it's really small, I could assume that your calculation is correct. Alright, then we can calculate the gamma score. So here I just calculated the gamma for C, right? And that's the 5th row in this table. If you want to calculate the last row, it's basically just 1 minus each of these values. So 1 minus gamma C1 to observe the gamma N1, and 1 minus gamma C2 to for the gamma N2. And then based on this, you could print all these gamma values. So we could interpret. Remember that we could interpret them as the chance of getting the correct prediction that if we predict this state as a CPG island, what would be our correct probability? So if we predict the first state as CPG island, the success probability would be 0.22, and so we would not predict it as a CPG island since it's less than 0.5. So we'd predict it as a from non-CPG island. And if for the second gamma, if we predicted it as a from a CPG island, the success probability is 0.6 or 8. So it's larger than 0.5. So we might have made the correct guess. So we would predict it as a from a CPG island. So do the same thing for all these 6 steps, and you would get the result for the forward and backward algorithm. Cool. Then similar things happened for the Viterbi algorithm. So remember that Viterbi algorithm actually is quite similar to the forward and backward, but they just do the optimization at each step. Try to remember the best path. So you calculated the sigma and the delta. Sorry. So this should be the delta and also the per se. And you compare each of them to do the termination. Remember all these results and to predict the result. So you can see that for this setting, the result of the Viterbi and the forward and backward algorithm should be quite similar to each other. Sweet, that's the first part of our lab. And the second part is to get an overview about the Cancer Genome Atlas public database. Well, this is a public database of the NIH, so it's TCGA. This part would be helpful for homework 6, but you don't need that for homework 5. So if you can Google it, you can go to the original website. They have the program's history or details and also publications related to them, and also describe of the whole database. You can check all this data type here. You got all the details about the file format and also the content. Cool. Just play around within that. So since it's a public database, a lot of labs has already developed some research tools to get access to the TCGA data, also for the R session. So for this lab, we are going to introduce a new a very convenient R software R package to get access to the TCGA data, which is the Firehouse. So if you can open our R session, please install the required packages. Well, you need the lemma to do the differential expression analysis, which we mentioned in the second lab, and also these two, the bladder batch and the SVA package, are used to remove the batch effects. And also the DevTools is actually helping us to get access to this Firebrowse R package. So we use the DevTools to help us install the Firebrowse R package, and we need the Firebrowse R package to help us to get access to the TCGA data directly in the R session. So if you can library all these packages, well, the dplayer and gplayer and ggplot2 are used for visualization. The GM and net are used for the regression. Alright, we have libraried them all, and now with the metadata cohorts, you can download all available cohorts. And if you want to check the original information, just the types of cohorts, and you can see the abbreviations and also descriptions about each of these cohorts. And then, how could we incorporate the information about all these patients? Well, in this package, by default setting, we can only receive 115 patients at a time. So I just created a script. So we just created a script for you guys to get access to all patients within one time. Actually, this code is inherited from the package's developer, and then we can check the dimension. So now we have 1,097 patients with 111 indicators. If you want to check get an overview of the database, we can go to this one. So the first column are the TCGA patient ID. Each row refers to one patient, and each column refers to a specific indicator, such as the age and also other things. And if you want to answer the question about such as how many of these patients are alive, and if you want to also get a hint about the distribution of the actual ages distribution, so we can calculate the percentage of deaths and living patients, and also calculated the ages mean and median value, and also the histogram of the about the patient's age. And then for the tumor subtype analysis, you can use the lemma to find differentially expressed genes. So please review lab 2, and you can assume that we have already performed the normalization and batch effect removal. So you can jump right to using the lemma. And we could also to do the clustering and the PCA to visualize the differentially expressed results. But before doing so, we have to remove some batch effect to help us to remove some batch effect. So we can use the Blender data to help us, and also the SVA package to help us to remove the batch effect, and also the combat to remove the batch effect. They are old friends, right? And use the lemma to do the differential expression analysis and select the top genes, do the k-means. Well, just pay attention. The log4 change I choose here is 2, and also the seeds for and also the k for the k-means clustering as 2 here. You might encounter some different value in your homework, and then run the PCA and visualize all these things. Oops, something bad happened. Cool. All right. Then you would get the plot as the group of non-cancer patients and the group of cancer patients. All right, that's all I have prepared for this lab. If you have questions, please feel free to reach me. I would hold the audition office hour this Friday, and just pay attention, the numbers using in this lab might be slightly different from the number in your homework. I wouldn't go through the part 2 and part 3 in the next lab, because in next lab I'm going to talk about the general procedures of doing a complete GWAS study, which is very useful in the statistical genetics. And if you have questions about that 2 parts, please send me an email. So good luck for your fifth homework.